Now, you would think, well, Bowie, why doesn't everyone have diabetes then? What is the thing that makes a diabetic's beta cells more susceptible than the next guy? So surprisingly, there's something going on where the beta cells of those with diabetes, well, beta cells in general, don't have high levels of enzymes responsible for buffering hydrogen peroxide. Or have they been compromised? Right? So part of the name of the game is twofold. It's you need to quickly neutralize the hydrogen peroxide to prevent it from degrading into hydroxyl radicals, as well as making sure that there isn't too much iron, free iron available to react with that hydrogen peroxide and devolve into the hydroxyl radical. Okay, so that's one thing. Like, you can see someone like, okay, why isn't everybody diabetic then? If you have these kind of two volatile components happening at the same time. So in the diabetic state, you have to ask, well, what has been compromised so that either A, there's an abundance of iron accumulating, or we don't have enough stuff to buffer the hydrogen peroxide. And so these are just three examples. There's more of them, but these are three enzymes within the cell that are known to buffer ROS accumulation. Glutathione peroxidase, hydroperoxide glutathione peroxidase. There's a whole slew of that GPX enzymes. There's GPX1, X2, X3, X4, as well as glutathione itself, which, if you're into supplements, you know that glutathione can be supplemented. But glutathione is a naturally occurring antioxidant in the body. And specifically for beta cells, glutathione is the tool of choice for neutralizing the hydrogen peroxide. Question remains, why do diabetics have low glutathione? Is it a game of, are we producing too much hydrogen peroxide, or is it a game of we don't have enough to buffer the hydrogen peroxide we have? It's probably a combination of the two, if you ask me, but that's what's going on. And so I've made a few TikTok videos recently about Sigurd Lenzen, who is the author here that I'm citing from his 2017 paper. And he has honed in on the specifics of beta cell ROS generation and buffering, and what parts of the cell are having trouble dealing with that ROS. So let's walk through this paragraph here. So at the top, in view of the low expression level of GPX1, which is the glutathione peroxidase. This is something that buffers hydrogen peroxide. So in view of the low expression of GPX1, in particular the beta cell mitochondria. So the mitochondria is low on this buffer for ROS, not a good sign. The regenerative reduction of oxidized glutathione achieved through glutathione reductase is crucial to avoid an overpowering of the limited antioxidant defense capacity. Okay, let that sink in for a second. We have mechanisms in place that are regenerating glutathione in order to keep up with this hydrogen peroxide creation. That is achieved through glutathione reductase. Okay, so that's just one of the enzymes that is going to basically repurpose glutathione to be used again. So this maximizes the chances for beta cell survival in situations of mitochondrial stress. Mitochondrial stress just means more ROS that needs to be handled by the glutathione. This necessity is supported by experimental evidence provided by studies in different animal models of diabetes. So in this DBA2 mouse model is a particular breed of diabetic mice. Now, this is a constitutive overexpression of manganese superoxide dismutase, which, if you'll remember, is the molecule responsible in mitochondria for converting superoxide into hydrogen peroxide. So let's take a second here. Do you remember this guy right here in the mitochondria? Superoxide, manganese, SOD, turns it into hydrogen peroxide. So this one particular paper, or this one particular reference here, is saying that they made a mitochondria in a diabetic mouse that made too much hydrogen peroxide. Okay. And this creation, when they made that enzyme that made too much hydrogen peroxide, it led to beta cell dysfunction. Okay, so we have some fingerprints there on what's causing this beta cell dysfunction. It's that overabundance of ROS. Now, below that, in the these are just different mouse breeds signifying the diabetic model. There's also another enzyme, nicotinamide nucleotide transhydrogenase. There's not a quiz at the end of this episode. It's just an enzyme. Prevents the prompt generation of GSH. GSH is glutathione. Okay, so in this mouse model, they took away the enzyme that helps make glutathione. And if you don't have glutathione, you can't buffer hydrogen peroxide. This showed an instantaneous regeneration of glutathione is mandatory to maintain protection of beta cell mitochondria, right? So they had this cell, they wiped out its ability to recreate glutathione, beta cell dysfunction. So again, more mechanisms, right? We've got too much ROS. If you introduce too much ROS, beta cell dysfunctions. If you take away the cell's ability to manage ROS, beta cell dysfunction. Okay. So let's do some speculation on our part. Okay, so think about this. In someone with diabetes state, they have, over multiple generations, they've been overstimulating their mitochondria, therefore creating a lot of waste, right? This could be overconsumption calorically, just in general, of food, but the processed sugar and those easily oxidized fatty acids, components of basically the standard American diet or the standard Western diet, where high energy creating a lot of waste. So our metabolism has been under a lot of stress for a long time. Combine that with poor activation of the enzymes needed to buffer the ROS. Remember, manganese, SOD, copper, and zinc, SOD, those are the enzymes responsible for creating hydrogen peroxide and neutralizing, like having that have someplace to go. So if we don't have those trace mineral components to allow those enzymes to do what they need to do, we're accumulating more ROS and not neutralizing them. Combine that with an overabundance of inflammatory amplifiers, iron specifically. I have focused solely on iron in my studies, but iron is in the cell in overabundance. We have more iron than we know how to package and we know how to get rid of. So it's floating freely in too many cells. It's floating freely in cells that are unable to handle the ROS that is accumulating in those cells. So you've got too much ROS and too much iron. The Fenton reaction, we are creating so many hydroxyl radicals, the cell is just overrun with oxidative stress. So let's walk through it. Beta cells always on. Now they're always on, for anyone that eats daily, right? That's kind of like a human thing. But typically in a healthy diet, you are eating nutrient dense food that is, yes, asking the cell to create energy, but it's also creating, or it's also supplying the resources the cell needs to get rid of the garbage that's accumulating because of that energy creation. So beta cells activated to the max all the time, creating unusual amounts of ROS because our foods have been modified in a way that we are just overridden with, processed carbs, processed foods, enriched foods, what have you. Meanwhile, they've been processed with all of those accelerants, and they've had the nutrients, the tools, the plant compounds removed so that we're creating a lot of waste and we don't have anything in place to neutralize the waste. So you've got too much garbage accumulating. Think of it like a hoarder's house. You've got too much garbage accumulating, and now there's iron hanging around because not only were those essential trace minerals removed from the processed food, we added gasoline to it by adding extra iron. If you check any enriched wheat flour, any processed good, chances are it's going to have reduced iron added to it. That is the match being lit and thrown into your beta cells, high ROS loaded cells.